are also we're also we're also very, very humbled by the support that Marlin and ALS United Ohio has given our program, both my laboratory and our clinic, and we're very, very grateful for that. What I'd like to do today is kind of two things. One, I'm going to sort of take off my clinic director hat and go a little bit more on to the lab side of things today, which I don't always do, and maybe go a little bit more granular. So I will try to put this into a context of why we're doing the things we're doing, but I thought it might be fun to kind of let you know some of the nitty-gritty that we're doing. And then at the end, put my clinic hat back on and talk about what is going on at OSU. It's wonderful to hear about Dayton with their clinic, and one of the big issues is access to multidisciplinary clinics, and this is just so amazing to have that extra clinic out there. There are research programs that I'll end with that I think we can all work together to try to get patients access to research opportunities as well, because that's a big limiter throughout the country for people. So I'm going to see if I can share my screen here without causing too much
I
problems.
I made you a co-host, so you should be able to, hopefully.
Yeah, it
You got
opened
it.
up.
Yes.
So let me see if I can get this open here. Okay. So now it's always a little bit weird to do virtual things. I am someone who loves to interact with my audience, and usually when I'm giving a talk, I can see people and how they're reacting. I get really excited about things, and so sometimes I start talking really fast. So Melinda and whoever else, you have total permission to interrupt me because I cannot see. And I'm just going to just go and be excited about what I'm doing. Now, hold on. Let me see if am I doing this right? Okay, hold on. Let's see if can you guys see the screen okay?
Yes.
Okay.
And
So it looks like I misspelled the word of there, which is a bad start, but let's get started. So one of the things that I'd like to kind of so my laboratory is really interested in why. Why is it that motor neurons in particular die when it comes to diseases like ALS? And one of the overarching themes that I like to sort of start with and step back from is the idea that neurodegenerative diseases like Alzheimer's, Parkinson's, and ALS are all, of course, terrible diseases, but they are characterized by very selective neuronal loss. In other words, not all of the neurons in your brain, the billions and billions of neurons, start to die all of a sudden. It's very selective. This is a slide that I like to use of PET scan imaging. And what that is these are just coronal sections of the brain where the yellow that you see are areas of hypometabolism, meaning that they're really, just think of it as the areas where the neurons are dying or sick. And on the left, you can see the initials of different neurodegenerative diseases, Parkinson's disease, multisystem atrophy, et cetera. There's Alzheimer's down there, frontotemporal dementia at the bottom. And I use this slide because I just want you to note that even though we call these all different diseases, which they still have in common a very selective loss or premature loss and dysfunction of neurons. And that's the mystery. Why? What is the cellular molecular basis of that selectivity? And motor neuron diseases are really where my laboratory and other labs like mine are really focused, because motor neurons are the coolest neurons. And they are also affected terribly in ALS and spinal muscular atrophy and diseases. And so this is just a review for everyone here. Everyone knows, I think, that motor neurons, of course, are those very incredible cells that connect your brain to muscle. they are extremely unique because most neurons connect to other neurons. And the upper motor neurons that are here in your cortex travel continuously all the way through the brain, all the way through the spinal cord, where they meet with the lower motor neurons. And in the middle there, you can see a little cross section of a spinal cord. And there is, I don't know if you guys can see my cursor, but there is a, actually, let me do this. I think maybe you could do, I'm going to use my laser pointer there. So you can see that there are the lower motor neurons here. Their cell bodies are there, but then their axons, the wires, that it is actually the shape of the cell is that it extends all the way out of the spinal cord into nerves and then deep inside the muscle where it makes this beautiful connection. This is an actual motor neuron connecting to one single muscle fiber, and this is that neuromuscular junction that you hear about. So I'm obsessed with these cells. These are incredible cells. Just the fact that they work at all is amazing. If you think about, this is an actual to scale drawing of a single motor neuron from a mouse. And you can see the cell body right here. You can see where it connects to muscle here. The muscle's not there. And what I want to emphasize to you is that the motor neuron is mostly an axon. It's basically a tube. It's this long continuation that goes out into the body. What is cool about this slide, this is a cell that has been labeled with a dye. So when you see yellow, all that means is that is the color of the dye. That's how they were able to kind of draw a scale diagram. But what they also use is a dye that stains for mitochondria. Mitochondria are those energy producing organelles in cells. And most, cells have, they have their mitochondria around here in the cell body and everything's cool. But motor neurons have way more of them. And also they have mitochondria out in their processes and way out here in the synapses. So in order to make that cell survive for decades and decades, then it really has to be a powerhouse cell that is making proteins all the time. So, it reads the DNA, turns that into RNA, and then the RNA is turned into protein. And that requires cells that are just cranking out proteins all the time. I'm kind of a nerd, and my daughters were going through junior high school like math. And so I calculated the surface area of a cylinder that is as long and thin as an axon. And it turns out that for one single axon that might go out to my leg muscle, for example, the area of that is visible. It'd be 6.28 millimeters square, which is a little bit smaller than a New York Times crossword puzzle square. So you're sitting there right now, your eyes are moving, you're breathing, you're looking around, you're maybe moving your body, and that is all dependent upon these incredible cells that you have in your body. So what I want to pivot to is what our laboratory is doing and kind of how the clinic and the laboratories set up with one another. You guys know that, of course, multidisciplinary clinics are where the rubber hits the road. That's where we care give. That's where we manage. That's where we try to do our best to optimize joy, minimize suffering. We have proactive care, and we have our dream team at Ohio State, and then of course the teams at the other multidisciplinary clinics that do just that. And then one of the things that Ohio State in particular has really innovated and led the country with is genetic testing and counseling. And you've heard me talk about the importance of genetics. And it's not just because we want to have therapies for people who have genetic disease, but also because it helps us learn. And that's what I'm going to talk about. That's what my laboratory does. When you have a gene variant, it allows us to build model systems. And those model systems in the laboratory allow us to understand what the hell's going on that's causing these motor neurons to die. And if we can do that better, then we can come up with better therapeutic targets than what have failed in clinical trials so far. And also, yes, targeted gene therapies for those specific forms that are going on. So this is a very important thing. It's something that I've really been very proud to be a part of building here at Ohio State. And it's really one of the things that does make us unique in this area is this connection that we have to the laboratories, not just at OSU, but throughout the world. So let me just give you a quick introduction, and reminder about ALS and genetics, and that is that the prevalence, of course, of ALS, how often that is occurring is about, now it's thought to be about 5 per 100,000, and whereas the incidence is lower than that. And one of the reasons for it is because of how compared to Alzheimer's, for example, it is more rapidly fatal than other diseases. In the genetics, this is an interesting graph that was recently put together. If you look at the outer circle here, the outer circle represents cases of ALS for people who do not have a family history. And you can see that in those people, we actually do find genetic causes, even though they don't have a family history. The inner circle represents those people who have a family history, and you can see that we have a lot more genes that are there. Here's a key of the majority of them, but of the main ones. But we don't know all of the genes, and it does vary by geoancestry, and there are reasons for that. So ALS over the last 10 years has been seen to be an increasingly genetic disease. I want to give a shout out to Jennifer Rogenbuck, who's not only a genetic counselor in our clinic, but is also, like me, a researcher, and she has written the genetic testing and counseling guidelines for the entire country. And these guidelines, by the way, have also been adopted by the European Academy of Neurology. And I just want to continue to shout that out because it's been right here in Central Ohio that this happened. This was not from Mass General, who we love and everything. This was not from Hopkins. This was not from the big places that a lot of people think about. This came right here at home. And we are the ones who led that. And it was all on the shoulders of Jennifer's work. One of the other things that I would like to, so this is a cool graph. Here's time. So here's the year. This is already, of course, outdated. And then these are the genes that have been identified. There's a lot going on in this graph, and I just want to use it to illustrate the number of genes and how rapidly those genes are being discovered that are associated with ALS. Just because a gene is associated with ALS doesn't mean it causes it. That's a little bit sometimes confusing. And that's something that my laboratory and other laboratories are very interested in looking at. But this is but what I want to do now is pivot to, yes, identifying a gene in someone may help with clinical trials, may help with clinical therapies. That's important. That's something that we want to do. But it also gives people like me an opportunity to understand the why. And we're going to talk today, just for about 20 minutes or so, about this KIF5A gene. So before I go and talk, I'd usually like to stop here. Are there any questions about what I've talked about so far?
If you have a question, feel free to unmute yourself and ask it, or you can put it in the chat. And Emily and I are monitoring the chat.
Are you okay? Good. And you guys are with me, I hope.
Yes.
Okay. So
oh, I think we might have a question.
Oh, good. Yes.
Question?
Yeah. This is John. I'm noticing some circles are larger than others. Is that related to prevalence or?
Yes. Awesome question. So let me go a little bit deeper here. So yes, the area of the circle is corresponds to its relative prevalence. So the C9 or 72 hexanucleotide expansion is the most common genetic cause thus far of both ALS in families and for people who don't have a family history. I should also note that this discovery also linked ALS to frontotemporal dementia, because this is also a cause of frontotemporal dementia. You can see here SOD1 happened to be the first gene that was discovered. And the blue is through what we call linkage studies. And this is what we used to do, the sort of the old-fashioned way, if you will, of finding a gene. Once you have a large family, you can sort of figure out what the gene is. But once the technologies changed and instead of, this is Sanger sequencing, means literally sequencing one gene at a time, sequencing the gene, sort of one nucleotide at a time. But then once it became possible to do parallel sequencing by machines that are capable of doing sequencing of the whole genome, for example, then that's when we started to understand these other ones. And then once we had large enough data sets from large groups, not just from a single clinic or a single family, but from large groups, then we were able to do association studies, and this is when we started to generate these more. So that's what the color coding represents. And that's one of the things. What OSU also and people with ALS right here in Central Ohio contributed to the discovery of KIF5A and other genes because of Answer ALS, another program that we had where people just, even though it was not a clinical trial, people selflessly volunteered to allow us to take their blood and to study them over time, even though they continued to decline, and that allowed us to make these discoveries, and that is something that is just an amazing gift. So
it does look like there is one question in the chat, and that question is, how can you have a genetic cause without a family history? Or did I misunderstand? Yep.
No, this is a great question. I think that we tend to think if there's a family history, then that makes sense. But gene mutations can happen de novo. They can happen just out of the blue, and that is one of the things that we appreciate from generation to generation. Most of those change, we call them variants in the genome. Most of those variants don't have a functional consequence, but sometimes they cause disease, and so that is just the randomness of genes being passed on, and sometimes a mutate. And there are places in the genome that where de novo variants can occur in a more likely way or not. These are all things about the genome that we're all still trying to understand and learn, but there are definitely hot spots in the genome where variants occur de novo, even though your parents don't have those variants. Another thing that I've learned from Jennifer and from understanding genetics more is that not just because you have an ALS associated variant in a gene doesn't mean you're going to get the disease. So for example, dad might have a variant that's associated with ALS, pass it on, and his son gets ALS, but he never gets it. So just because you have a variant, there is uncertainty when it comes to these things. And that is one of the things from a clinical standpoint that can be very frustrating. But it is also something that is trying to teach us something. And that's what's important. And in fact, the KIF5A story that I'm about to tell you is a good illustration of that. Good question. So let's pivot now to what we do when we find KIF variants so KIF5A variants were discovered. This is a fun cartoon because this is the KIF5A protein, this orange protein, and the function of KIF5A. Remember that long axon that I drew, that was illustrated earlier in the talk. In order to get proteins and other things to walk all the way down that axon, because they're made in the cell body, but they have to travel all the way down that axon, you have to have what are called motor proteins. And KIF5A is one of the major motor proteins in motor neurons. And it literally has a domain here, a protein domain, that sticks on top of the railroad with the microtubules that make up the axons. And then it attaches to things. In this case, this is a lysozyme that is a large sort of balloon that is carrying cargo down the axon, and that is its job. That's KIF5A's job. So here's another sort of image of it. And one of the amazing things about the KIF5A protein, and again, I'm not going to go too deep unless you want me to, is that it has the three very distinct functional domains. And now this is sort of speaking my language when it comes to the lab. I'm very interested in how alterations in proteins like this cause disease, and in this case, motor neuron disease. Here's another way of looking at a protein. We call it a domain structure. And what you can see is there's a domain here, the motor domain. This is this foot domain right here. You have a stalk domain that is right here. And then you have this globular domain, which is right here. And that's the one that kind of connects to different things. One of the amazing things about KIF5A is that variants had already been associated in the feet that are associated with different kinds of diseases, but they are all diseases that affect motor neurons, but they're not ALS. Hereditary spastic paraparesis can be a very disabling disease. Axonal neuropathies are also very disabling. Not life-threatening, but nonetheless disabling. But what was really the big deal was that these two papers came out in 2018 that described variants associated with ALS in KIF5A up here. And I was very attracted from a biochemical standpoint and just sort of why is it, again, that you have variants here that cause one disease and variants here that cause another disease. ALS here, motor neuron diseases that are not ALS here. And so again, these variants are what opens up the door for us to start to ask questions in the laboratory. And this is the paper, by the way, that we contributed to, and people here in Central Ohio contributed to with their genomes that they donated for this paper, along with about 1,000 other throughout the country that donated through that Answer ALS. So I'm going to kind of blow through this slide pretty quickly, but what I want you to know is that the variants that are associated with ALS result in a change in this globular domain so that it no longer sort of has the same surface to bind to things. And one of the things that other people had discovered, if you look at this normal picture of KIF5A, here's walking along the thing. Here's a cargoes just like we were drawing. One of the things that it does do is it can inhibit itself or bind to cargo. But in the variants, it actually, what people showed in cell cultures was that it did not go in this auto inhibition and it walked really fast along the axons, and that is what was thought to cause the toxicity. We call that in mechanism of disease work, we call that a gain of function. The ALS associated variant made this protein change its function into something that was toxic. And in this case, in cultured neurons, it made it walk faster. Does that make sense? What we wanted to do was, this is a fancy slide that says that I'm going to skip the slide actually. One of the things that we wanted to do was to create then an animal model. These are some variants that are associated with ALS, and all of these here with the red, these are just biophysical properties that show that compared to the wild type human, these have big changes. And what we wanted to do was create a mouse model. And so this is how researchers will try to understand. We'll make human-derived stem cell cultures from patient cells, and we'll try to create an animal model so that we can see what the consequence of that variant is in an animal. And so we did that. We were the first, and I think so far the only group in the world that has made a knock-in mouse model of this. And by knock-in, what I mean is that we took the mouse genome and we literally, using CRISPR-Cas9 technology, which you guys may have heard of, but that gives us the ability to actually go in and change a single nucleotide in the DNA of an animal or in cell culture. And Mehmet engineered that to make mice that have that knock-in mouse model. This is a slide that basically shows us that we did what we wanted to do. And we have animals that have all wild type KIF5A. We have mice that have one gene copy that is wild type and one gene copy that is mutant. And we also have actually homozygous mice, which means that they have 2 copies of the mutant form of ALS-associated ALS. And so all you need to know from this is that we actually have 3 different types of mice in the lab that we created. One of them is normal mice. One is mice that have 1 copy of wild type, 1 with mutant. This is what, by the way, people with ALS have. They have 1 wild type and 1 mutant. And then we also we're able to make mice that have all of it. This is the one of the first things that we found, which was a little bit abnormal, and that is that you guys know that the genes make proteins, and I just showed you that cool stalking protein. This is one way that we in the lab we sort of take spinal cord or we take brain from the mice after we sacrifice them, and we are able to quantify how much is there using antibodies that are very specific for that. And what we found actually was that these mice actually had less of the KIF5A protein than expected. They had the same amount of RNA that we would have predicted, but there was something wrong with their making of it. And so what we actually find, this is just another sort of way of looking at this. We found that the heterozygous mice and the homozygous mice have less total KIF5A protein. So think of it as, instead of there being something wrong with the protein, we actually find that these animals have less of the protein that we expect, which is a first clue of what's going on. When we looked at these mice, did they have ALS? The answer is no. They ended up surviving just as long as wild type animals. So we did not model degeneration in these animals. This kind of bummed us out a little bit at first, but to be honest, there are very few mouse models in ALS that have degeneration unless you overexpress a toxic protein. We generally mice are not a great model for ALS, but that is one of the main ways that we try to look at things, and we can come back to that question in a bit. So one of the things that we did is and this is all done, of course, very humanely with anesthesia and so forth. But one of the things that we wanted to ask is, okay, these mice don't have a degenerative disease, but are they able to repair from injury as much? Because these are proteins that walk proteins down the axon. And so here's our paradigm. We actually take mice, adult mice. We perform a surgery where we open up the skin on their leg. This is the sciatic nerve, this white thing. We can actually do a very controlled crush procedure that results in them being paralyzed in that limb for about 2 months. But by 2 months later, they are completely healed. And to be honest, you just would never even know that they had a problem. Mice are pretty incredible when it comes to healing in this model. And so this model is a completely reversible paralysis of this leg. It's a very well-established model. But what we did is we did electrophysiology. And Kelly Rich here, who was a graduate student in my laboratory, now a postdoctoral fellow at Harvard, and also a genetic counselor, by the way, she did longitudinal studies of these animals after the nerve crush. And this is just a look. This is an anesthetized mouse. And many of you who are patients, who are people with ALS who are watching right now, have had the EMG test. Well, this is a mouse having an EMG test. And they are having the same kinds of shocks and the study for their CMAPs and their munis. And this was developed here at OSU with my colleague and friend, Dave Arnold. So now, all I want you to do here is just look at the fact that this blue line goes down and then comes back up. This is the recovery of the electrophysiological measures in a mouse, in the motor neurons of a mouse. This is weeks post nerve crush. And so there's a total loss of the ability of those axons to work. And then there's a regrowth that occurs and a normal functioning that occurs. Pretty cool. But what we found is that these animals that have the KIF5A variants, they did fully recover from their injury, just like the wild types, but they lost motor neurons. Their motor neurons did not. And this is motor unit number estimation. And they actually had fewer motor neurons motor units that were there. And so what this is an impairment in the ability of the KIF5A variants to repair. And this is just a reminder that that corresponded with there not being enough of that. And so we concluded from this, and just sort of just give you a flavor, that we didn't have a neurodegenerative phenotype, but there was impairment of motor unit. There was no evidence of aggregates that people saw in cell culture, but we did see a reduction in the total amount that's there. And so that is what we were looking at. And we actually went ahead and looked at other mice that had already been established by others that don't have any KIF5A. And we found that they actually were very similar, that those animals also had a difference in their ability to recover. And so just to kind of really not get too much into the weeds there, because I think I already have gone too much into the weeds, that what this shows is that the ALS associated variants, we believe, maybe in cell culture have this increased processivity, but what we find in an animal model of those is that there is less binding capacity, and maybe that there's an impairment to recover. And this is a paper that was published last year by Kelly. And it positions us, really, this is a starting point. Now that we have this animal characterized, it's a starting point to try to ask some questions about vulnerability. And this gets back to what I was talking about before. Just because a gene is associated with ALS doesn't mean that that variant always results in ALS or causes ALS. But what it could mean is that it's a risk variant. And one of the things, that I hear all the time, is, people who have had some kind of injury, or they have had some kind of surgery, or they have had just wear and tear throughout life. Could it be that having specific variants of some of these genes puts you at risk, so that starts the process of ALS? Is that something that puts you at risk, of having that? And that's something that I think I find very interesting and compelling, and something that we are very much wanting to look at. This is just a quick shout out to the people who are currently in my laboratory. I'd like to give a shout out to Megan Baird, who's my postdoctoral fellow, and also the recipient of the first ALS United Ohio research gift that I am extremely grateful for and humble for, and want to thank Marlon and her entire team, and of course all the people who donate to ALS United Ohio for this incredible gift, because this allows us to do this work. And it is really a privilege to have this community here in Central Ohio who's focused all in this. Here are some formal laboratory members and collaborators. Here again, my thanks to ALS United. We're very lucky also to have grants from the National Institute of Health, ALS Association, and others, and also very generous donors throughout the years who have paid for fellowships and just basically are pipetters and little things in the lab that add up to big things. So before I go on to the clinical trials stuff, are there any questions about the KIF5A story so far, or anything? I wanted to sort of illustrate. I can talk more about what we're planning to do if you'd like, but I thought that that would be a good way of just sort of sharing what the so what answer is to so what if we have discovered a new gene. This is one sort of tangible result of that.
And if you have a question, feel free to either unmute or type it in the chat and we can read it.
I was just going to comment, Dr. Kolb, that if someone asked me to put in a nutshell what you just explained, there is no way I could do that. However, seeing it and seeing the slides and hearing you talk about it, I feel like I understand just a little bit more. And those two things probably don't make sense. I can't explain it, but I do feel like listening to you talk about it and looking at the slides, I feel like I have a slight better understanding of a tiny little amount of information about ALS research. So thank you.
Well, I appreciate that. I mean, I think that, I mean, it's hard, and it's a moving target. And I think, even, for that's what excites me on the research side is that it is constantly growing and learning. But it's hard because it's not just a simple nutshell thing. I think if I were to put one thing, one sort of take home point is that genetics in ALS is not just relevant for people with a genetic form of ALS. I think that people who do not have a genetic form of ALS, I think sometimes feel that this doesn't mean anything to me. And that's what I want to try to change that idea. The sort of this era of genetics in ALS is teaching us fundamental things about ALS, about why the cell selectivity. And I think that is going to benefit everybody, in different ways. I guess that's the nutshell, is that the genetics benefit everyone, even if you don't have a genetic form of ALS. So, at ASU, OSU, we really try to have a sort of I like to call it the dessert tray, that comes by in a fancy restaurant that has all these amazing looking desserts, and in clinical trials and projects, because I think that we get approached by a lot of companies, a lot of different groups, and I make an assessment based on sort of how they fit into our portfolio, so that we have a good diverse range of options for people to pick. The first sort of fundamental one, of course, is that we are a leading Healy platform trial site, and one of the leading recruiters in the entire country, in fact. That platform trial has been sort of at a standstill with respect to regimens now for many months, but there is a new one coming soon, and so we're very eager to get started on that.
Dr. Kolb, sorry,
yes,
can
ma'am.
you explain what a Healy platform is? Because we hear that term a lot, and I know what it is because I looked it up, but I think a lot of people don't understand what the Healy platform, what that means.
Oh my gosh, yes. I'm so sorry. I would have put some slides in there before to do that, so I apologize that I didn't do that. But yes,
No, that's okay.
what makes the Healy platform trial unique is that it is a consent to a platform, which I'll define in a minute, that brings the ability to test, and by test I mean testing whether or not agent A, B, or C is effective to slow the progression of ALS faster than if you were to do single clinical trials by themselves. We actually have a link to a video on the Healy platform trial on one of our things that we can send, put in the chat. And Cheyenne or Carly, if you're able to put that link in the chat while I'm speaking, that would be amazing. Sorry to put you on the spot there. But the Healy is the name of a very generous donor who donated, I think it was something upwards of $50 million to Merit Sokovich and her team at Mass General and their wonderful ALS program. Merit is one of my friends and mentors and colleagues, and what she did with that money was create this platform trial. And so what it allows, normally a clinical trial, a standalone clinical trial, has a placebo arm, and that is essential. No one likes that, but it is essential in order to be able to know whether something works or not. What the Healy platform trial does is, of course, it also has a placebo, but because it's a shared placebo, in other words, what you do is you consent to the platform, and then you are randomized to agent A, B, or C, which is pretty amazing. And so what that means is that that you don't know whether you're going to get agent A, B, or C, but because you are randomized to it, you actually can have a greater chance of being exposed to drug rather than placebo because of the way the platform trial works, because you're randomized to different things that's there. That's probably not a great way of explaining it, but there's a video. We actually developed the video that is being used nationally. Our fingerprints are all over this thing. So I think that so that is something that is really a way also, by the way, for small biotechs to get their agent into an ALS clinical trial. So, you know, big, powerful pharmaceutical companies, they'll just go ahead and pay for their trial. That's okay, fine. But there are lots of exciting possible ALS studies that from new biotechs that are out there that would never see the light of day if it were not for the Healy platform trial. And there is a scientific review board and, you know, all of that kind of stuff. So it's another way to get, I mean, basically shots on goal. That's the thing. We need more clinical trial shots on goal, and we need to do it in a way that is efficient, in other words, faster. We need to get to the answer faster. And we also need to try to contend with this idea of exposure to placebo. Have to have it, but how can we limit that as much as possible? And that is something that the Healy platform trial is one of those solutions. In addition to the platform trial, we are a part of currently a part of a gene therapy study. So in this case, yes, this is a study that is really reserved for people who have a specific gene mutation. And I get that, you know, does not apply to the vast majority of people, but it is a very important shot on goal for those patients. And if it works, it's something that, of course, we will learn from, very much like the Qalsody SOD1 gene therapy that is now FDA approved. And so this is part of that era that I'm talking about of, you know, here where we have the full circle going up to specific gene therapies in that case. We have two clinical trials on deck. And there's actually more than that, but there are two here that I have. One of them is a study that is going to be a classic sort of randomized double-blind placebo-controlled study of this is a monoclonal antibody that is against a protein called TREM2. TREM2 is a protein on microglia. Microglia are the neuroinflammatory cell within the brain. And so this is a study that I'm excited about because it is tackling the neuroinflammatory side of ALS. And that is something that we are in the process of getting ramped up. We have been selected as a site, and I don't know what other sites there are. We might be the only one in Ohio. But we are not yet activated. I don't think any sites are activated for this study yet, but hope to be sometime in the fall. And then there is actually another type of gene therapy for SOD1 patients that we will be a part of in the future. And so these are the clinical trials that are coming up. I also want to finish now on another really important thing that I know that also has been a part of in helping to enact, but this is really now at the fundamental grassroots, you know, sort of effort that here, OSU, once again, we are going to be the site in the Ohio River Valley and in this area for this project. And I really want to advertise this now because this is something that no matter what multidisciplinary clinic that you receive your care at, we really want to get the word out that this is something that you can participate in. Of course, any of our clinical trials, but this is one where there really is no exclusion criteria. So back in 2021, Biden signed into law the Act for ALS. I'm sure you guys have heard about this. I'm sure Marlon and her team have talked to everyone about this because this was a really major thing. one of the things that did was it required an expenditure for ALS research by the FDA and the NIH, and that resulted in this program called Access for All in ALS. And what that means, not just access for multidisciplinary care, but actually access for research. And that is something that really is a problem for people who not only do people not have access to multidisciplinary care with their travel as their mobility decreases and so forth. And what they did is really over $38 million was spent to create an infrastructure of 2 hubs and 34 spokes. You think of a wheel. Those hubs, one hub is Mass General, the other one is the Barrow in Phoenix, and there are 34 spokes, clinics that are expert clinics in clinical trial recruitment and execution. And we are one of those spokes, and we're very, very proud of that. This is something that's just started. In fact, today we were activated to recruit patients for one of the 2 of these, and we're about to be in the other. So I'd like to take about 5 minutes to go through this, because this is something that I really want to get on everybody's radar, and we'll have more information on this coming soon. This is what they gave us, and it's kind of a rough draft, and so I just wanted to share it anyway. But this is some information that we can also make available. I'll have my slides available for you all and send these to Melinda to share to everybody. But this is something that, again, an NIH expenditure that is coming here to Central Ohio. So your taxpayer money coming back to Ohio. And that is something that has these projects, and all ALS Assess, we hope to enroll nationwide 1,600. Look at that. That's more than has ever been done. And also control participants. I'm thinking caregivers here who will consent to a program of study over time. And I won't go into details of that. I think that probably deserves a separate sort of presentation at another time. And then another one is called ALS Prevent. And I have this thing for ALS Prevent. This is a very unusual study, which is separate from Assess. It's unusual because we're actually looking to recruit people who are the first-degree relatives of people who have a genetic form of ALS. And again, I know I harp on the genetics here, but one of the things that this study does is we're not, in this study, not following people with ALS. We're actually following their first-degree relatives. And it is a very exciting way to look at, can we identify the reasons that people get ALS or don't? And can we also identify people before they get ALS? In other words, can we see what it is that turns them from not having ALS to having ALS? This is being done right now in a number of areas in specific ways, but this is the first time it's been done in a big national way, and we're very excited about it. I'll finish by just saying that just a couple months ago, there was an additional article from the National Academy of Sciences, Engineering and Medicine. This is independent of what I just told you. And what they did is they actually, this is something that I encourage you to go and look at if you haven't. I'm sorry for this very long thing, but you can probably just Google this. But this is a publication that is free to everybody to look at. And what they recommend is additional expenditure for specifically ALS to build out this multidisciplinary care. And what this means is that like Alzheimer's, that has Alzheimer's disease research centers throughout the country, and that allows centers to have this sort of combined model of a multidisciplinary clinic that is integrated with researchers who are studying that, much like what we have already been doing here at Ohio State. This is something that is now possibly going to be funded in the near future. And of course, we are going to work very hard to be one of those centers for this region of the country. But this should also bring more money into all of our multidisciplinary clinics here in Ohio. And so this is something that's very exciting. this is my sort of standard shout out that, that we are in an exciting era, even though we don't have anything that, that in my back pocket that I can just give to everybody right now. There is reason for enthusiasm and investment in research to push things forward. And that is because of this genetic understanding, the modeling, which I hope I showed you today as an example of what we're doing in our lab. And then the fact that we can actually alter gene expression in motor neurons in humans. This is based on the success in spinal muscular atrophy, and also, I think, a qualified success in ALS with the SOD1 is bringing us to where we really believe that even for people who don't have a genetic form, our understanding is going to allow us to really have effective treatments. I'm going to end here by showing Cheyenne Grove and Carly Failer, who are our rock star research coordinators at Ohio State. Here is us a couple, I guess a weekend ago or so at the walk in Columbus, where we had a lot of fun and we were able to distribute some of our research information. If you have information, any interest or questions about Seth, Cheyenne and Carly are the ones, and this is our research email that we have dedicated to all of that, and phone number. So questions about participation, just in general questions about what's available, please know that we also will happily refer you to other clinics that have trials that we happen to not have. But we are unique in having some of these natural history and science-based ones, and of course also the Healy platform trial. So please don't hesitate to reach out to them. They're very responsive, and we are very interested in sharing our information. And so hopefully I can't see you, so I can't see if everybody is yawning or has completely fallen asleep, but I hope that was intelligible, and I'll stop now for any questions.
Thank you. That was amazing. I agree with Marlon. I don't know that I could sum it up, but I understood a lot of what you said. So I just loved it. So there is a question in the chat, and it said, hold on, it says, I know you mentioned that just because you have a genetic variant doesn't mean you will get ALS, but are there any triggers that they are fairly certain of, in either familial or sporadic?
I mean, this is a great question that I think we all wish we had the answer to. There are variants that it seems like you're absolutely going to get it. There are variants that are much more dicey, where it's not clear whether you're going to get it. That gets started. That uncertainty is really in the wheelhouse of what genetic counselors like Jennifer have to contend with. I'd also say that that's a dynamic and changing thing, so that a variant that might have been reported 10 years ago that was sort of uncertain significance, 10 years later, it might be found to be actually very pathogenic, because we are learning from people who have these in real time. So my guess as far as this conversion, so and this is how I think of it, is that, definitely the main number one risk factor for ALS is aging. And I think that the process of maintaining the biological task of maintaining that cell that I illustrated for you, just go back and imagine, or extend your arm right now and move your index finger up and down, and look at that finger way over there moving, and imagine that there's these microscopic thin cell axons going all the way through your arm, from your neck all the way out to your finger. Imagine the biological task of keeping that alive for 100 years. I mean, I'm pretty good with houseplants, but I can't keep them alive for 100 years, where our bodies have to keep the motor neuron alive. I tend to think that when you have an ALS-associated variant, that sets you up to as a risk factor, that the normal aging process, in some way, there's some point where the cell just says, I can't do this anymore. And that's the beginning. That's when the cells begin to die away. And I think that there's probably, I believe, and you know, I think others believe this, is that there's something that seems to initiate the process. And then there's another thing that maybe is related or maybe not, that is associated with the progression of disease, because ALS definitely starts and then it just happens. It just sort of seems to roller coaster down. And I think that I think of it as dominoes that are all set up, and there's something that hits that first domino, and then there maybe is something else that is set up where those dominoes just continue down. And so I think genetic variants, like for example, KIF5A, if I had a KIF5A associated variant, maybe I don't have enough KIF5A to carry things down my axons during periods of stress or during periods of injury, which is what our animal model shows. We've actually used it in a chemotherapy model, and in mice that get chemotherapy that have KIF5A variants also don't heal as well. So could it be that these KIF5A variants mean that things are pretty normal, but then if something happens, or if you have an injury, or if something, let's say you have a viral infection, and then things start to go downhill. And that's what we don't understand completely, but I think we are getting there.
And then there was one other question in the chat, and I think I need a little bit of clarification. The question itself is, will I ever speak again? And what I'm guessing that means is there research into the reverse of symptoms once you have them?
Right. Yeah. No, and I think that, you know, with my clinical hat on, that's one of the hard truths, is that we don't have things to reverse the loss that's occurred. This is one of the things that, you know, I think that doctors sometimes, who aren't me or ALS doctors, I think doctors are sometimes, it's hard for them to say those words. But the fact of the matter is, we do not have the ability to make things that are lost come back. I believe that 50 years from now, when we look at the history of medicine, we will have therapies that will be able to prevent ALS from happening first, or at least stop the progression from happening. I think that those kinds of wins will happen. And then later on, I think we'll be able to have better technologies to completely restore and repair. I think that's another hurdle, I think, scientifically, that we're come at. But that's my guess. That's only my guess. I'm not God, and I don't know the future for sure.
All right, if you have a question, feel free to either unmute yourself or type it in the chat.
Yes, Ray. I had the question about the trigger, but my other question about the two, I guess, is I know in the past I've seen some things they suspected, some chemicals, things of that sort, extreme athletes. Does any of that stuff still hold true, or they've been able to prove that that's not the case?
Definitely, the data. So there's two things. A lot of that is based on what's called, epidemiological data. Epidemiological data is sort of looking at large populations, seeing what they have in common, and trying to come up with conclusions based on that. And it's hard, because humans are very heterogeneous. They're spread all out. We don't really have, unlike the animals in my lab, I don't know all of my exposures. Like when I walk through a golf course, I am getting exposed to pesticides that were just sprayed on that golf course, but I don't know it. And I don't know all of those things. And so I think that that's hard to do. So there's no hard data that says that this or that chemical causes ALS. We do know that in general, people who are exposed to things are more likely to develop things like Parkinson's disease and other neurodegenerative diseases, and ALS is one of those. As far as the athletes piece, I definitely think that there's something to that. The one piece of data that still sticks out is that people in the U.S. Armed Forces are twice as likely to get ALS as those who don't. They tend to be robust, athletic people who get through their military training. They tend to be men. and men tend to have a little bit of a higher risk than women. And so one of the things that is always remarkable to medical students who follow me into clinic to shadow me is how otherwise healthy ALS patients tend to be. They tend to not have a lot of these other diseases of middle age. They tend to be people who are athletes when they were young. They tend to be robust. And I wonder if the same genetic sort of gifts that people have that allow them to be robust from an athletic perspective, is that also, are there maybe similar things that put people at risk? I don't know that, but I'll tell you, having had two OSU and pro football players who had ALS, it really convinces me of that link with athleticism and ALS.
Thank you very much for the answer. I always find this, I have ALS, but I do enjoy the science behind it, hoping that someday they'll find a cure, but it's interesting to me.
Thanks, Ray.
Yep, thank you.
I'll put out a final call for questions.
Well, this has been really wonderful, everybody. And thank you again for this forum. And then also just thank you so much for the trust and the sort of the gift that you've had in us. It's very important to me that when people donate money, that it doesn't just disappear and fluff away, into this abyss, and we really here at OSU want to be very accountable. And a lot of the things that I described today are things that really would not have happened were it not for people who trusted us to work and try to understand. And that really is what it's all about, as we make progress so that we can have something in the clinic that works.
Thank you so
Thank
much.
you, Doctor. Oh, sorry. Sorry, Melinda.
No,
I was
that's
just
okay.
gonna
Go ahead.
say thank you so much, Dr. Kolb, for being here with us tonight. And we were very excited to be able to make that our very first ALS research gift to your lab. And it's exciting to hear about the things that you're working on. And we always like having you, and we are excited to get to partner in a lot bigger way more recently. So appreciate you being here, and Carly and Cheyenne as well.
Absolutely. I just want to remind you that if you